Okay, so we started. Welcome to everybody to a new webinar for the International Legume Society. Happy to see you here again. We are here with Professor Yongfeng Ai from Saskatchewan, so Canada, University of Saskatchewan, Canada. He is an expert on carbohydrate in general, carbohydrate coming from plant and crop processing and everything that comes from extrusion and all the utilization. And today he's going to talk us about starch utilization and how it's intertwined with pulse processing. So, Professor Yangfeng.
Thank you, Nelson, for the nice introduction. I would like to thank the International Legume Society and the President, Professor Tom Workington, for giving me the opportunity to share our research in Canada. Like before we start the webinar, we had some quick chat. We had about half meter of the snow over the weekend. And today now, at the current moment, the temperature is minus 30 degrees Celsius outside. So I'm very happy we were able to manage to make the webinar today. We are going to talk about post-processing and the starch utilization. Here's the agenda of my talk today. I will just use one slide to talk about my research program, carbohydrate chemistry and the utilization. Let me just bring the, I will just use the mouse then. To talk about our research program, to look at carbohydrate chemistry and the utilization. After that, I will give you some background information regarding generating new ingredients from pulses, and also some of the unique functional property of pulse starch. After that, I will share with you one ongoing research program in our province. We call it the Strategic Research Initiative Program. Usually we call it SI program. So for this program, we receive the funding from our provincial government and also the federal government in Canada to look at the pulse starch utilization. I will give you a program overview. After that, I will highlight some of the most exciting research from the different projects in the program. For example, the clean label modification of pulse starch and also the strong gelling ability of pulse starch for biomaterial applications. For my research program at the University of Saskatchewan, we have four research themes. The first one, we look at the carbohydrate modification and the utilization. For example, the pulse starch chemistry and the utilization actually fit into this theme. Also, we look at other crops that may have a starch and other functional carbohydrates. For example, we look at the oat starch and also quinoa starch. Saskatchewan is also a main producer of those crops in the country. The second thing, we look at carbohydrate nutrition, which we look at the development of resistant starch, resistant maltodextrin, dietary fiber, and also low glycemic food to improve the health of humans and also pets and animals. The third research theme, we try to use a novel processing method to process different pulses and cereals we grow in the province in the country. The last one, we look at the biomaterials. For example, I'm going to talk about the strong gelling ability of pea, lentil, faba bean, and other pulse starches. So we try to use the strong gelling ability of the pulse starch to develop some gel for like as a sensor, and also for packaging application for drug delivery use. So these are the four research themes in my program. Pulses actually is a very important group of crops we grow in Saskatchewan, in North America, and also in the world. Pulses, they are defined as the dry edible seeds of crops in the legume family. They are different from soybean and the peanut. The main reason is because soybean and the peanuts, they are primarily grown for the oil extraction. And also, pulses are different from fresh peas and the fresh beans. According to what I learned from Dr. Workington, these are usually considered as vegetables. In North America, including Canada and then the States, the pulses we grow mainly include pea, lentil, dry bean, chickpea, and faba bean. In the past 10, 15 years, pulses actually have gained a lot of interest from the food industry for the ingredient innovation. On this slide, I would like to use pea as an example to explain how the processing is done. Usually, we start with the whole seed. The first step, usually what we will do is the dehulling. So after dehulling, we have the split pea, and we also generate the hull fiber. Actually, hull fiber is a very popular fiber for human food application and also for pet food use. When we have the split pea, we can use two methods to fraction the different component inside the pea to a different ingredient. First thing we can do is actually we can use a milling, just a very simple and straightforward method. We can produce the pulse flour, which has a very good nutritional value and a very good functional property. We can use the dry fractionation as well. We usually start with the pea milling followed by air classification. This is the most common method we used in Western Canada. And then by using this approach, we can obtain the starch-rich flour and also the protein-rich flour. Also, we can use a wet fractionation method to produce the starch and the protein of a higher purity. We can obtain the isolated starch. Usually, they have the starch content higher than 96%. So for the isolated starch, some of very classic examples of to use them in the food industry include like the Chinese vermicelli or glass noodles, and also the gummy bear candies, as you can see here. They are based on the strong gelling ability of the starch. For the protein isolate, actually they are considered as a high value ingredient in the food industry now. They are very common to use as a main ingredient for plant-based meat and also for plant-based beverages and even for protein powder. So these are really how we can fractionate into the high purity, the starch and protein. Actually, another ingredient usually people kind of ignore or didn't pay too much attention is you also generate the cell wall fiber from the wet fractionation. Actually, this fiber has some very unique property as well. They have very fine particle. And sometimes this kind of fiber also called inner fiber or cotyledon fiber. So I hope this gives you an idea of regarding how we can generate new ingredient from pulses. When we talk about the ingredient fractionation, usually we need to look at the chemical compositions of the raw material we use, right? So regarding the pulses we use for the ingredient production, as I just covered. So actually, when we look at the chemical composition of the like a pea, lentil, and the faba beans, you can see the starch content of this crop, they usually range about 40 to 50%. For the protein, it usually range about 20 to 32, 33% at the high end. And also they are known as a good source of dietary fiber, usually about 12 to 20%. But the key thing here is pulses are different from other crops in the legume family we use for the production of a plant-based protein. For example, for soybean, after they become mature, the seed usually have less than 1% starch. They may have some other carbohydrate, but the starch content is very low. But because of the chemical composition of the pulses, based on this chart, you can tell if we produce about one volume of the protein, usually we will generate about 2 volume of the starch. Actually, according to the information from the industry, sometimes they may even generate 2 to 3 volume of the starch as a co-product. So the challenge for the pulse industry is actually not just in Canada, in other part of the world as well, is how we can generate the value from this co-product. Because remember, when we produce 1 volume of the protein, inevitably we will generate the 2 or 3 volumes of the starch. So this starch can come in 2 forms. One is the, as I mentioned, from the dry fractionation, we can have the starch-rich flour. usually they contain about 60 to 80% starch. And we can also have the isolated starch. They have a much better purity. Usually we look at more than 96%. So when we talk about the utilization of any ingredient, we usually need to look at the unique functional and the nutritional attributes of this ingredient. So I would like to use this webinar to share with you some of the unique functional property of pork starch. But before we go into that topic, I would like to use 3 slides to share with you some general information regarding starch chemistry. It is known starch is a bipolymer of amylose and amylopectin. You can see on this slide, the starch, they are present at the granular form in nature. And then the starch, they consist of the amylose and the essentially linear amylose, and then the highly branched amylopectin, as you can see on the slide. So actually, because of the different the structure of the 2 molecules, the amylose content actually really affect the functional property and the nutritional value of starch. The amylose content of starch can range from 0, such as in waxy starch, to 85%. Some of the high amylose starch can go up to this high level of the amylose. And then the amylose content can really influence the thermal property, pasting property, gel formation, and enzymatic hydrolysis of starch. Since we talk about the functional property of the starch, I want to use this slide to go through some of the key concepts of a starch and also the instrument we can use to analyze those different properties. As I mentioned, starch exists as a granular form in nature. So when we soak or we suspend the starch in water, actually it can absorb about 30% of the water. And then the granule will also swell to a limited level, and then we have the swollen starch granule. But this process is reversible. So which means is if we use a low temperature, a mild condition to gradually dry the starch, actually the starch can go back to the native original form. However, if we heat the starch in the presence of water, and then the temperature go above the gelatinization temperature of starch, starch will be gelatinized. Actually, we do this every day in our kitchen because when we try to cook the starchy food like pasta, noodles, and baked bread, and many bake the potato, many other food, we try to gelatinize the starch. After we gelatinize the starch, we continue to heat and then to stir the sample. Actually, we will obtain the starch paste. So for these 2 properties, like starch gelatinization and starch pasting, they are usually connected. Sometimes you don't see them separate. Usually for the gelatinization of the starch, we use the instrument called the differential scanning calorimeter, DSC, to determine the gelatinization properties. But for the starch pasting property, is usually about the development of the paste with a good viscosity. For this property, usually we can use a rapid viscous analyzer, RVA, or we can use a rheometer. For some of the starches, for example, pea, then lentil, faba bean, and the normal maize, and then normal wheat starch. Actually, after we cool it down, the starch paste will develop into a viscoelastic structure with no fluidity. It cannot flow. It can hold the structure. So usually for this kind of structure, we call it starch gel. And then another like a good example for this is like it's not like the Jello product we can find in the market. It's not. I want to make it clear it's not made of starch, but that kind of structure is how we define the starch gel. I'm going to show you more photos. And then for the starch gel, if we heat it in water, and actually can go back to starch paste. So this step is also reversible. For us to determine the strength of the gel, actually we can use a texture analyzer, but definitely we can also use a rheometer to determine the rheological property of the gel. Then after we have the gel, when we store at a different temperature, particularly at 4 degrees Celsius, actually starch will go through retrogradation. retrogradation is usually considered as undesirable because they will cause syneresis. Another word for syneresis is expulsion of the water from the food matrix. So you will see the separation of the water from the solid, and also retrogradation is partially responsible for the staling of bread. So most of the time for the food application, we want to control the starch retrogradation. I hope this slide gives you some idea regarding how we study starch as a very fascinating polysaccharide. Also, starch is a very important component in the human diet. So after we ingest the starch in our food, I will go through hydrolysis. It is known this hydrolysis is usually completed by 6 enzymes. They are salivary alpha amylase, pancreatic alpha amylase, glucoamylase, maltase, sucrase, and isomaltase. So after this hydrolysis, the starch will be converted into glucose. Then glucose can be absorbed as a very important energy source for our body. But in all the different starchy food, actually there's a fraction of the starch cannot be hydrolyzed even at the end of our small intestine. So this fraction of the starch is known as a resistant starch. So resistant starch can be passed into our large intestine for microbial fermentation, and then the short-chain fatty acid will be produced as one of the main product. And short-chain fatty acid, for example, acetic acid, butyric acid, and propionic acid, they will provide the different health benefits to our colon. So they are usually considered as a desirable. And very interestingly, some resistant starch, they can even survive the extensive fermentation in our large intestine. They can be secreted into our feces to increase the fecal bulk. So actually, the resistant starch, because of this nutritional feature, actually some of the resistant starch, they are accepted as a dietary fiber, even according to the latest definition of a dietary fiber given by FDA. So after sharing with you some of the background information about starch chemistry, I want to share with you some of the unique functional and nutritional attributes of pork starch. Actually, in 2019, the first MSc student in my program published this paper in food chemistry. In this paper, we characterized pea, lentil, and faba bean starches and compare them with the other starches available in the market as a very important source of the different starch for various applications. So I just, in summary, when we try to compare the different pork starches with waxy maize, normal maize, and tapioca starch, pork starches, they show a high amylose contents. This is a very interesting feature of a pork starch. The pork starch usually have about 38 to 41% amylose content according to iodine calorimetry method we use in our lab. But other starches like those listed in our paper and some other like a potato and many other starch, they only have about 1 to 31% amylose. Because of the high amylose content and also the long branched chain length of the amylopectin, actually pork starch, they are usually known to have a greater retrogradation rate. So if we want to use a pork starch as a thickener or as a binder or stabilizer in food, they are usually considered as undesirable because it will cause a syneresis and also the quicker staling of the product. So it's usually considered as undesirable. But for other food application, retrogradation may be a good thing. And also the starch can form very good gel. I try to explain in the jello, even though it's not a starch-based product, but let me just use this slide to give you a better idea regarding the strong gelling ability of pork starch. You can see on the left, we have the pea starch gel. This is after cooking, we cool it at room temperature for 2 hours. You can see the structure, it has a good viscoelastic property and it cannot flow. By contrast, when we look at the tapioca starch, you can see actually after the cooking, the paste can flow. They have very good fluidity. So these are the differences between pork starch and many other like the commercial starches. So why this is important? When we look at some of the application of starch in the market, actually the food industry already make a very good use of the strong gelling ability of the pork starch. I already show with you the example of a glass noodle, or another we call the Chinese vermicelli. You can see this product. Actually, in some part of the like some regions in East Asia, what I learned is actually some residents, they will consume these noodles as their staple food. So they actually consume a large volume of the starch over there. But unfortunately, in North America, we don't eat so much, even though we can find this product in local restaurant. Also, in some local market in East Asia, I learned is the starch can be used to prepare, we call it jelly type of appetizer and dessert. So this is actually a product prepared from pea starch. So you can tell it has some similarity to jello, right? But another thing I want to share with you is we have to consume this product fresh. Again, this is related to the high recirculation rate of pork starch, because if you cannot finish this product on the same day, you leave it in refrigerator, second day when you wake up, you want to eat it, you will see a lot of water will sit at the bottom. This is because of the recirculation and the syneresis. And also, the pork starch, they are known as a good substitutes for the gelatin for the gummy bear candies, and also to replace pectin, because both of them are used for the gummy bear candies. And even the pharmaceutical industry, they have developed some very good starch from the chemically modified pea starch. Actually, they can use it to prepare the softer capsule shell, as you can see here. So actually, they can be used to replace gelatin, because this can be a good option for vegetarians. And also sometimes pectin, they are more expensive. So the chemically modified pea and other pork starch can be a good replacement for both of the two ingredients. After we talk about strong gelling ability, I also want to mention that the pork starch, they have a greater enzymatic resistance in a raw granular form. This is important for actually the like the feed application of the pork starch or pork flour we produce in Western Canada. But another thing I want to share is after we cook the starch, I'm only talking about the starch, not the pork seed. If we have isolated pork starch and isolated waxy maize, normal maize, and tapioca starch, based on our data, at least in vitro, their digestibility is very similar. Another very important piece of information I want to share with you is, according to my working experience with the pork starch in the past 8 years, very interesting is like the pea, lentil, and faba bean. No matter where we obtain the starch, we have worked with the starch from Canada, from the States, and also from Europe. Very interesting is these pork starch, they seem to belong to one group. And they are very different from the starch from other crops, like maize from potato or tapioca, oat, many other crops. So what I want to share with you is, since we are talking about the value-added the application of the pork starch, if we find one good technology for pea starch, I would like to say most of the time, the same technology can be utilized for lentil, or they can be utilized for faba bean starch. You can achieve the similar R&D purposes. With
Sorry, but you are muted.
Can you hear me now?
Yes, now we can.
Oh, sorry, I didn't do anything. I don't know what happened. I hope. Okay.
Just a click. Okay. Now
okay. Yeah, I
start
can.
from. Okay,
Okay, I can
from this slide.
From this slide, right? We're okay.
Yeah, sure.
Okay. Yeah, I can. For some, all of a sudden, I can also use a laser pointer. Let's continue with the SI program. And for this program, we focus on pork starch utilization. And the program, for this program, receive funding from our provincial government and also federal government through the Sustainable Canadian Agriculture Partnership. So for this program, we work with Dr. Tom Workington and Dr. Buyamin Tarrant. They actually provide us the high quality policy from in different market classes like the pea, lentil, faba bean, and the wrinkle pea, and others we include in the project. So the first thing we actually try to do is we want to improve the dry fractionation and the wet fractionation method we currently use in the market. And actually, in the interest of time, I'm not going to go into that, but we did develop some very good technology that can extract 18% more protein in different varieties through the dry fractionation. And also, one of the master students in my group, she tried to control the different temperature and also pH during wet fractionation. We can obtain the isolated starch and the protein isolate of very good quality. And very interestingly, because of the different temperature and also different pH we use for fractionation, the obtained ingredient, they also show very diverse functional property and very diverse nutritional value. I think even just through the wet fractionation approach itself, we may be able to generate different ingredient without further modification. And after the fractionation, we can have the starch-rich flour and also the isolated starch. So what we try to do is we try to use this ingredient to develop novel food ingredient. For example, resistant starch, resistant maltodextrin, dietary fiber, the pre-gelatinized starch. And also we work with microbiologists to use a fermentation technology to convert, particularly for the starch-rich flour, to convert it into like a prebiotic, probiotic, and also symbiotic. And we also work with engineers to develop the biomaterial and also biomedical materials from the isolated starch. Because you don't want to use the starch-rich flour for this application, the main reason is, as I mentioned, the starch, this ingredient only have about 60 to 80% starch, so which means you still have a protein and a fiber as impurities. So we can only use isolated starch for these materials. So for the whole programs, we have 9 research laboratories in Western Canada to collaborate to look at a different aspect of the research as I just shared. We have Dr. Tom Workington and Dr. Buyamin Tarrant. They are breeders. They produce the like high quality and also the new varieties of the pulses for the different research purposes. We have a group of food scientists and microbiologists, including myself and Dr. Mike Nicholson. Actually, he also gave a webinar on the same platform back in December 2023. And Dr. Darren Colbert and Dr. Takuji Tanaka, we are in the same department. We also have animal nutritionists, Dr. Denise Bolio and Dr. Daniel Clumbers, and then they look at the health benefits of the resistant starch, resistant maltodextrin, and the dietary fiber we generate from different crops, and then they look at the health benefits in animal models, in particular in pigs. Also, we have engineers, Dr. Bishnu Acharya and Dr. Malcolm Xin, to look at the material science side. In the last part of my webinar today, I want to share with you some of the most exciting findings from our research. The first one is we look at the clean label modification of the pork starch, because nowadays consumers usually they don't want to have a chemically modified ingredient added into their food. So we try to come up with a clean label modification for starch and protein and many other ingredients. So one method is an enzymatic method we can use, and also we can use a physical method. For example, like an extruder. Actually, we have this lab scale extruder in our lab. And the first study I want to share with you is we use a very specific type of the alpha amylase called mutagenic alpha amylase to enhance the functional properties and reduce the digestibility of a pork starch. So for this study, we use this enzyme in abbreviation MGA. We try to modify the pea and the faba bean starch, and then the normal maize starch was included as a control. And then after the hydrolysis for 24 hours, you can see actually this enzyme hydrolyze P and faba bean starch in a very similar pattern, but it's very different from a normal maize starch. In brief, this enzyme actually hydrolyze the P and faba bean using a surface pitting, a surface erosion approach. So because of that, you can see after the hydrolysis, the surface of the starch granules become less smooth. And also, you can see the different small pieces, and also there are some fissures on the surface of the starch granule. But for the normal maize starch, this enzyme hydrolyzes through an inside-out pattern. So because of the pattern, you can see they have all the different holes. These holes go all the way to the center of the starch granule. Actually, after this modification, one very interesting result I want to share with you is you can see at a significantly reduced the retrogradation of all the starches. For the P starch, it reduced from 56% to 16%, and then for the faba bean from 46 to 14%. Then for normal maize, it reduced from 35 to 1.5%. Another very interesting is when we look at the resistant starch content, actually, this modification can significantly increase the resistant starch content of all the starch samples even after cooking. You can see for the P, the resistant starch content doubled. Then for the faba bean, it tripled, and then for the normal maize, as also triple. As I already shared with you, the definition of a resistant starch. Actually, if we look at the carbohydrate nutritional value, this starch, the modified starch, they will have a better nutritional value. And this is for the enzymatic modification. And actually, Dr. Tom Workington and I, we are co-supervising one PhD student to look at how we can use extrusion as a clean label method to modify starch. This study is actually funded by the same funding program, but under a different project, like we call it the ADF program, and then the research is not from the SI program. But the very interesting result is when we have the native P starch, and then we use the extrusion to modify it, and then we dry it under a controlled condition, and then after that we mill it, we can obtain the modified starch. This is considered as a relatively simple, and then definitely they fit into the clean label ingredient modification. So what we found is when we look at the pasting property, so for this figure, you can see is actually we cook the starch all the way. This is a temperature curve. We cook the starch all the way to 95 degrees Celsius, then we cool it down. Then we observe the viscosity change. These two are the viscosity curve. What we can find is actually after the modification, the viscosity of the P starch decreased. So we are thinking, okay, when we look at the granular structure, this is the granular morphology of the different native and the modified starch under SEM. We observe the loss of the granular structure. It doesn't have the native starch structure. So we are thinking, yeah, this makes sense because of the loss of the granular integrity. We don't have such a high viscosity. But actually, when we try to cook the starch to 120 degrees Celsius, so this is we use as a condition to represent the high temperature processing. And very interesting is for this modified starch, when we cook it to 120 degrees Celsius, particularly after we cool it down, you can see the viscosity is actually higher than the native starch. So this gives us two very interesting things. One is if we want to use the starch for high temperature processing, this starch may have some unique feature because it can better retain the viscosity even you cook it to 120 degrees Celsius. Another very interesting thing is based on the comparison between 95 and 120 degrees Celsius, seems like there's a stronger entanglement between the starch molecules. So which means you need a higher temperature to separate them in order for them to develop viscosity. So we are thinking this kind of molecular entanglement may make the starch less digestible. Actually, we did the in vitro starch digestibility, and then we cook the starch. When we look at the digestion curve, actually this starch indeed show a greater reduction in the enzymatic hydrolysis. So when we look at the resistant starch content, we can increase the resistant starch content from about 2% to 5.6%. And then for the last part, I would like to share with you how we try to use the strong gelling ability of the pea starch for biomaterial application. So for this ongoing research, actually we obtained the pea starch, we blend it with the PVA and then water and glycerol as a plasticizer. We also add some salt. Then we cook everything in one pot. And then after we cool it down, actually we are able to obtain very interesting conductive hydrogel. So you can see this gel actually has a very good flexibility and ductility. So when you look at the, like we try to even put a weight to stretch the hydrogel, and actually you can see it has a very good flexibility. And then another thing I want to share with you, the thickness of the gel is only 1.5 millimeter. So actually it's not a very thick gel. When we try to use a dynamic mechanical analyzer, actually this gel gives us almost 300% tensile strength. Another very interesting of this type of gel is it has very good anti-freezing property. When we have the same gel, we store it at a minus 20 degrees Celsius for at least overnight. Then we take out the gel to determine its different mechanical property right away. There's no thawing or warming it up back to room temperature. There's no such thing. We did the measurement right away after we took it out from the freezer. You can see it can retain the flexibility and also the ductility. And then the strain only decreased about from 290 to 250 5%, we can retain the tensile strength. So because of the very interesting property, we are thinking to use this type of gel to develop some strain sensor, particularly at a very low temperature, like in the winter in Canada. If we can develop some useful sensor for this, this will be very meaningful. And then also Professor Malcolm Xin and the postdoctor fellow in his lab at the University of Manitoba in our neighboring province, they try to use the pea starch to develop the sponge. And then this is a video. And then actually they try to put the sponge in water. They can absorb the water. Let me just share with you the content. You can, I hope you can see it. So what they try to show here is after they compress the gel, this is different. This is not the, just give me 1 minute. Let me get the laser pointer. Then, so they try to compress it, but after they put it in water, they can absorb a large volume of the water. So they are thinking to use this as a material for hemostasis material. And this is another material. They also use the pea starch to produce the sponge strip. And then this has a very good flexibility. Let me just share with you the video. Oops. Just give me 1 minute. Yeah. So what I want to share with you is this material is actually made of 100% from pea starch. There is no other biopolymers. Definitely you have to add some water, some glycerol as a plasticizer, but there is no other biopolymer or any other polymer. So they are thinking to use this material as a bandage for bandage application. So I think that's pretty much all the like research I would like to share with you in our program at our university, how we work together with the experts in different fields to find a way to process pulses, particularly the pulse starch with significant value addition. I would like to use this slide to just share with you some of the most exciting findings. actually starch is a very common and abundant polysaccharide. So when we talk about the utilization of any starch, we need to look at the unique functional and nutritional attribute of this starch. And then enzymatic and physical method can be applied to diversify the functional property and increase the resistant starch content of a porous starch. And also the development of the novel biomaterials based on the strong gelation capability of porous starch. I think this area is very interesting, and we will try to secure more funding to look at the future opportunities. In the end, I would like to acknowledge the financial support from the different funding agencies in our province and also in our country. Without their support, we couldn't get the research done. This is a photo of our group taken in 2023. So majority of the work was done by Fang, who is here. Fang is a PhD student co-supervised by Dr. Tom Workington and myself, and she's going to defend in next few months to graduate as a PhD student. And also Chen Yong is a current PhD student in my group, and he's the person really work a lot with the starch-based biomaterials. If you have any questions, you can send me email. We can continue our discussion. I think that's all for my webinar, and thank you so much for your attention.
Thank you, Yongfeng. Thank you. It was very interesting and a pretty novel topic for our series of webinars. Now, if there's any question from the group, I'll be able to take them directly or in the chat. While people is thinking about the question, I have a couple for you.
Sure.
So, first of all, well, this is clearly from my background because I come from breeding, and one question would be, were you in the condition of characterizing some population and finding differences in varieties or lines of PE or other pulses, or you just took like a big bag of seed of seeds and pea seeds and stuff like that, because obviously a lot of people here is also doing breeding and variety improvement. So that's the topic.
Yeah, for sure. Actually, for us, we just look at the starch. You can just go to the market and to grab anything that is available. Actually, one advantage of the researcher in my department is actually we have a crop development center, which is also in our college, like Dr. Walkington and Dr. Terry and many other excellent breeders in the same college. We work very closely to look at the variety difference. Actually, it's not just they will be the same. No, they are very different. And actually for the SI project, because I didn't have all the time, right? So like I can give you another example. Dr. Tom Workington and Dr. Terry, they are working together to breed some high protein pea. And we are thinking if we, in the raw material, we increase the protein content of the pea and other crops, maybe during the fractionation, we will obtain the more protein in the protein-rich flour from the dry fractionation, and then also like the wet fractionation, we'll get more of the protein, and also maybe easier to fractionate the starch. The reason I say that is actually because of the faba bean in nature it has more protein, and because of that, during the fractionation, we feel it's actually easier to extract the protein from faba bean in comparison with a pea. So that's what we found, because you can easily separate them. Another thing is we need to pay attention to the genetics and the environmental those factors. You cannot just ignore them. Oh, they will be the same throughout the year. And actually, the climate is changing, right? Then there are many other challenges we are facing. So with the advantage of like the problem development center, we usually call CDC and like the breeders in the same college. We actually sometimes we also look at how we call it G/E effect. So we look at both factors, then how they will interact to affect the starch and protein component. So I think what I want to say is for the even just for starch utilization, you cannot just look at starch alone. It's only one of the component from the very high value seeds we grow in Saskatchewan in Canada. So we have to look at them as a whole, like a whole seed. And you need to look at the protein, you need to look at the varietal effect and many other things. So I'm glad I'm part of the community and actually we're working together to look at the different components.
Okay, thank you. It was a complete answer. I got a question from the chat. It says, "What is next for your research on pulse starch?" So something about the future.
Hi, Sherilyn. Thank you for your question. Yeah, I can definitely share more, like based on our current collaboration and also the different projects. I think what we are thinking is for the food application will still continue because of if we look at the global starch industry, like global starch market, actually what I learned is there's a more interest in using pulse starch to replace the other starch in different application. It's a good thing for the industry. One of the main reasons is, let's say 10, 15 years ago, because of the volume of the pulse starch in the market is so small, because back to then, we didn't fractionate as much protein as we are doing now. And now we also produce more starch. So the food industry actually, for certain application, they want to use more of the pulse starch. So I think we will continue to look at the food application of the starch. And then definitely, as a one research group, we are more interested in link the structure with the function with the nutritional value. We want to just to cover all of them, not just, oh, we have another type of starch. It's more, I think we need to develop more resistant starch to look at low glycemic benefits of the process. So for food application, I think we will continue in the research. Another very interesting thing is, as I shared on last part of the slide, is like for some of the material application, I think they are also very interesting, particularly if we can use the starch to prepare some gel. And then for some of the new application, even for like agriculture, for like, for example, one example I can share, like a release of fertilizer. We want to try to look at all the different aspects, because we now know the starch, the pulse starch, they can form very good gel. So can we use them for the material application? Another thing I want to emphasize is actually, like the material we are developing, they will have very good biodegradability. They will be highly biodegradable because of the nature of the material we use. So I think that may also, in the long term, may have some benefits. Another area we are thinking is really to use some fermentation technology. Fermentation is a very popular word in the food science field now. We are thinking to use more fermentation technology through working with the colleagues in our department. Also, like our protein researcher, Dr. Mike Dickerson, we're going to work together to, for example, for some of the like the starch-rich flour we produce in our province, can we use some fermentation technology to increase the protein content, to drive the microorganism to utilize the starch more efficiently? So that's another area I think we are very interested. We're very interested. One last thing, as I already commented, is I think sometimes we don't just look at starch alone. Some of the research we're working in the lab now is actually we realize if we can enhance the interaction between starch, then to enhance the interaction between starch and the protein and the fiber, and actually we can produce some very novel ingredient with a low glycemic benefits. We have one, the doctoral research fellow in our lab, Dr. Yi Kairen in our lab. He's trying to develop some whole cell flour. We try to use some novel technology to just isolate the whole cell, and then this can be a novel flour. But at the meantime, we have a high protein, high fiber. So we can, yeah, those are some of the, I think the most promising directions we're going to pursue in next few years.
Okay. Thank you again. Do we have any other questions from the audience? Meanwhile, just a small curiosity, one of the two projects that you showed, the gel that was also shown to be torqued, etc., you presented as conductive. Is that correct?
Oh, sorry. Yeah, because of we haven't started like to measure the conductivity of the gel or use it as a sensor yet. But on my slide, there's one part I didn't emphasize enough, but we add salt into the gel as well. So the salt, actually we use a quite high concentration. For some of the gel, we even go to 13% of the salt. Okay. So because of that, in nature, it will have the good conductivity. But definitely, as I share with you, is the project is still ongoing. But to share with you the strong mechanical properties are the main purpose today. But we are pretty sure because of the high salt concentration, and they will have very good conductivity for sensor application. Yep.
Okay. Very well. I guess that with this, we are going to the end of our webinar. I thank you again, Professor. The webinar is going on our website and on our YouTube channel very soon. Thanks again. It was a pleasure to talk to you.
Yeah. Thank you so much, Nelson. Thank you, everyone.